Yeah, welcome, those of you that are awake. This lecture will be from Carolina Wählby, who is working with image analysis and deep learning. And she is she's located in Uppsala at the SciLife lab. And so please go ahead.
So, thank you, Daniel, for inviting me, and thank you all for joining this morning. So I'm going to talk about image analysis and AI in microscopy-based life science research. And what you see here in the background is the output from some of our recent projects. And actually, half of the images is fake and half of the image is real. But I will get back to that later in my talk. So my plan for today is to start to talk a little bit about why we would want to use the combination of microscopy and image analysis. And then start with some traditional approaches. That's really where I come from. I've been working with image analysis for many years, and also 6 years in the US at something called the Broad Institute, which was all about large scale, high throughput screening and using image analysis to understand drugs and the interaction of drugs with cells and model organisms. And then sort of tell the story about our discovery of deep learning, or how we started to use it and how we're using it today. I will also talk a bit about digital pathology, which is a really fast growing field, and say where we're now and where I think that we will be going in the future. And I'll talk about training, because I think that's one of the really key points in any deep learning, how you train your algorithms. And also a bit about in silico labeling and denoising. I'm not going to go into details of the networks and their architectures for two reasons. One is because that would take too much time from all the things that I want to say, but also because I'm not really the expert of these things. It's very much driven by many of my PhD students. And also what I've seen over the years is that there are always new versions and new approaches coming up. And very often it's more the training data and the evaluation and the sort of different pieces together that make it successful or not, and maybe not actually what the exact design choice that you make. And I'll be very happy to answer any questions at any time. I love to be interrupted. And I will try to keep an eye on the chat, and also try to keep an eye on if you raise your hand. And if you want to, I'll be happy to share my slides after the lecture. So let's get started. And the most important thing is the people that are working with this. And I just wanted to put them here and also add them in the end. This is my group in Uppsala. And these are the people that would know each and every detail of everything that we did. And the group as a whole works with image processing analysis with applications in biomedicine and life science. I don't have a single microscope in my lab. All the data comes from a vast number of really excellent collaborators. So what we do is everything is computational, and our goal is always to sort of increase the scientific value of the images that we work with. And it's 2D, 3D, time lapse, and a lot of what we do is now based on deep learning. So why microscopy and image analysis? Sort of the hypothesis that we always start with is that some kind of model system that is modeling life in some way. It could be modeling the human body, or it could be modeling some kind of interaction between, for example, a drug and the response in a cell or something like that. In all of these model systems, our hypothesis is that it changes its morphology, it changes the way it looks in response to some treatment. And our hypothesis also that these changes in morphology can be imaged, and they can also be quantified by digital image processing and analysis, so that we can get a number out of our experiment. And this could be a range of things. This is one example. These are cells that grow on the surface. And the blue things here are the cell nuclei. And the green is this protein that is in this case present in the cytoplasm surrounding the nuclei. And in this case, the green has moved into the nuclei in response to treatment. And this would be the kind of thing that we could image and then quantify. And this is then the response to a cancer drug. This is something that I spent a lot of my time on when I was working in the US. It's a little worm that is about 1 millimeter long. It's called C. elegans. And we use it to study fat metabolism. If you can see the red stuff here, that's the fat. And in this case, the fat is all over the place, while here it's located to small patches within the worms. And actually, just last week, we were ready with the publication of this, showing how the same genes that affect the metabolisms in humans also affect the metabolism in these worms. And that drugs can then be tested on the worms to see if we can influence the metabolism and then influence how fat is stored also in humans. This is an example of zebrafish that we use to study drug effects and how they affect embryo development. So in this case, we want to measure the change in morphology. So all this are things that I will get back to. And here's more details of what's happening inside the brain of other model organisms. And just to give you a kind of feeling for the power of this image to the right here now is an image from a larger scale project that we did some years ago together with AstraZeneca. And they create new molecules that could potentially be used as drugs. And they then grow cells. And in this case, it's breast cancer cells. And then they add different stains that stain different things in the cells and different proteins that are involved in sort of the cytoskeleton and also the cell nuclei. And the reason why these images that all show the same cells and all show the same stains look so different is because these different drugs affect the morphology of the cells. So there's some names on some of the treatments here. These are a lot of drugs that are used to treat cancer patients, but there are also some that say AZA, AZC, AZJ. These are all the new secret compounds of AstraZeneca. And what they want to do is to see, are there any of these compounds that have the same or similar effect on the morphology as the drugs that we already know? And by using imaging, you can screen hundreds of thousands of drugs very quickly and without needing any use of animals or humans. You just work with the cultured human cells. And you can very quickly get a feeling for if these new molecules have the potential to be used as a replacement or as a complement to already existing drugs, just based on their similarity in how they affect the morphology of the cells. So image-based methods, and also now I talked about drug screening, but images are also used in diagnostics. And this is not a new thing. Imaging has been used for a long time to look for example, changes in morphology of cells in relation to cancer. This, what you show here is how cells from the cervix change as cancer is developed. And this was, of course, for many years observed visually and is still observed visually in many countries. But already in 1956, people tried to automate this process. This is the cytoanalyzer from 1956. It was created before there were digital cameras. And this was actually one of the first systems that had a color screen and could create images. And it was called Excel, and before the more well-known Excel that you heard about. And it was created by Evert Bengtsson in Uppsala in 1984. So really pioneering automation of image-based screening. And I borrowed these images from Evert, and he was also the supervisor when I did my PhD. So then, if we think of all these images and these changes in morphology that we want to measure, I would like to start to present like the traditional approach to sort of go over to why we're working with deep learning now. So in a simple task, like here, the goal is to measure the size and the shape of each of these cells. And here the cell nuclei are blue, and the borders between the cells are green. It's a stain that stains the sort of the junctions between the cells. And this is fluorescence microscopy, meaning that we image each of the fluorescent channels in a separate image. So the blue is the nuclei, so we can separate that image, or we can have that show that image separately, showing only the cell nuclei, and also the cell junctions as a separate image. So the obvious first thing we would want to do is to find the cell nuclei. And since they're brighter than the background, we can do a simple thresholding. But if we do that, you will realize that some cells are not properly separated. And in this case, the colors mean nothing but the fact that it's separate objects. So these objects are still connected and these are still connected. So we need to do something more to separate the individual nuclei. So then we can add some additional knowledge. We can use watershed segmentation, which separates everything by thinking of it as a landscape and then looks for individual little mountains in this landscape. But obviously this doesn't work on the raw image because there's too much structure. We get over-segmentation. So then we can do some smoothing to reduce the over-segmentation. And it does help a little to separate some objects, but we still get some over-segmentation that we don't want. Then we can play a bit more, and then we can say, okay, let's suppress local maxima that are in close proximity and then apply the watershed. Then we solve some of the problems, but a few new ones appear. Then we can maybe approach the problem by looking at shape and apply a distance transform and then the watershed. But then again, the cells are not perfectly round, so we still don't get a perfect result. But then if we combine this distance transform and the suppression of local maxima and the fact that local maxima have to be far apart, and then also the watershed, then we can get something that is more or less mimicking what we would do if we would do it manually. So we get a more or less perfect result. Then the next step, once we have these segmented nuclei, we want to find the borders of the cells. So then we can use these as seeds in this image and apply a watershed. But again, we don't get a good result if we do it right away. Then we can use a different version of the watershed, and in this case, something called a gradient weighted seeded watershed, and we get a fairly good delineation of the individual cells. But what I wanted to realize from this is that a really simple task, such as defining a cell, may really require significant parameter tuning and also some expert knowledge. And the same is actually true for the next step in this kind of classical approach, that now that we found the cells, we wanted to do some measurements. And then we also need a bit of engineering to really design meaningful measurements. So if you think of this whole process with this conventional image analysis workflow, is that you would start with some input image. You very often have to do some pre-processing, maybe compensating for variations in illumination or reduced noise. Then the next step would be the object detection, which I just described in the previous slide. You would want to do some feature extraction, and then you might have some conventional machine learning approach to make your decision about, for example, healthy or sick looking cells. But the whole process requires a lot of thinking in every single step and a lot of engineering. And as you have learned now during this course, it would be quite different if you would use a deep convolutional neural network, because you would instead start with manually annotated data saying that, okay, these are the healthy looking cells and these are the sick ones. You would do the training and then you would continue with using the network for classification of previously unseen input images to get your output. And really what's inside here is then just all these different filters, more or less, and different steps that are learned during the training process. And in many cases, these filters will be very similar what you would have designed if you would use the original approach. But really the big difference is now that the features are not designed by the human engineers, but they're learned from these large amounts of training data. And I will get back to this really the large amounts of training data. And as I mentioned before I came to Uppsala, I was at a research institute in Boston called the Broad Institute. And there we worked a lot with image-based screening. And when I was there until 2015, we had not really started to use much deep learning. But then as I came back to Sweden and I had this really talented group of PhD students, and they were starting to say that we should really look into this. And more people were realizing that, you know, cats and dogs could be classified really well with deep learning. And microscopy images are in some sense easier than images of natural scenes. One thing that you can see is that if you have a single microscopy experiment, all the images will be of the same resolution. I mean, the cats and the dogs here, they can be imaged at very different, either very close or very far away from the camera. But here we always have the same resolution. We also have the same perspective. We always see the cells as they are growing on the surface or of these tissue slices that they are just this thin slice from something three-dimensional. If the imaging has been done carefully, we have similar illumination and color, and we do not have any typical orientation. So in some sense, it seems quite reasonable to describe a microscopy image content by a series of convolutions, and maybe more straightforward to do so than compared to, for example, cats and dogs. But my dear friends in the US, they were really skeptical to deep learning. When I said that I wanted to try these ideas, they said, no, that's just a hype. Don't do this. Deep learning requires enormous amounts of manual annotated training samples. There's no way you can get that in microscopy. Deep learning is a black box. We don't know what really happens. We really need to have full control. You can't use these things. And it requires tuning a large number of parameters, and it's really more a craft than a science. So don't go down this path. And I didn't really listen to what they said, very much because of my PhD students. In October 2015, this paper by Olaf Rönnenberg and the first description of the UNet came out. He used it in the ISPI tracking challenge. And working with images of cells and looking at this image here, visually we can see the outline of the individual cells, but solving a segmentation of these unstained cells with traditional approaches and sort of trying to describe it mathematically is super difficult. And when I saw these results, I was really, really impressed. And then one of my students, Sajid, he came to me and he said, now I I managed to do this the exact same thing as Olaf did on our own data. Can we buy a GPU cluster? And this is what we did. And this is really where our journey to deep learning and image analysis started. Because seeing these results really convinced me that we could go beyond what can be done with traditional approaches that I had been working with before. And next, I want to show you one of the problems that we approached, because it really taught me something that I had not really thought about before. And this was a problem that we had solved once before, so it was sort of revisiting an old problem. And in fact, it was actually one of my other PhD students who said he was half a year away from his defending his PhD thesis, and then he said that he wanted to do deep learning. And I told him that there's no time, you have to write up your thesis. You can't start a new project at this point. But then he really wanted to do it. So I told him that if you write your thesis really fast, you can do some deep learning, but only if you finish your thesis fast. And that was the best carrot ever for anyone writing a thesis. He wrote it super fast, and then he got to spend the rest of his time on this project. So what is this? And this is what is called a multi-well plate, which is a piece of plastic with little holes in it. And in each of these little holes, we have these zebrafish embryos. They are about 2 millimeters long. And what we use them for is to study drugs that may be dangerous for embryo development, and then dangerous for patients that are pregnant. So what we do is then to add drugs to the wells, and then we want to have an automated way to see if the embryo development is affected. And in this case, our collaborators told us that the effect that we're looking for is the bending of the tip of the tail. You can see in these fish, the tail is straight, and here we see a slight bending. And as I said, we had already approached this problem once before. And then we used the classical approach of first finding the well, and then finding the fish in the well, and then finding the tails of the fish, and then measuring the curvature. That would be like the classical approach with quite a lot of parameters to tune. But we got a good result. But then Omer, my student, he wanted to apply the deep learning. So this was one of our really first experiences with a larger scale experiment. So we trained a neural network. In this case, it was early in time, so it was an AlexNet. And we had 50 images with fish that were treated with the drug, and 50 wells with untreated fish. And we did augmentation by flipping and rotation, and then we had 28 independent test images. So it's quite small data set. And then, of course, we wanted to determine the probability that previously unseen fish were classified as deformed. So I'm showing the results now only on 4 images, but then we had a much larger data set for the evaluation. But what we saw then was that if we had the treated fish, we had a high probability of this deformation of the fish. And for the untreated fish, we had a low probability of deformation. And in this case, we didn't say what is a well and what is a fish. We just threw in the images as they were. And I was still a bit skeptical to this deep learning and wondering, and maybe it's not the fish, maybe it's something going on in the image background that sort of decides the deformation probability. How can we know that it's really this fish tail that is saying that we have deformation or not? So to really understand that, we got 93% accuracy. But we couldn't be sure that it was the fish and their bent tails that were really learned by the network. So I made the students manually with like Photoshop go in and delete the fish in the images to see what happens. So if we input images without fish, we get no deformation. So the presence of fish is required for the detection of deformation, so that's a good thing. It would have been really suspicious if it could detect deformation if there were no fish in the well. So that was a good, but we could still not really know if it was these bent tails that were learned by the network. So what we did next was to manually delete everything but the tails of the fish to see what happens. So we fed the network only with fish tails. And the classification failed. So we thought that we had created a network that could detect bent tails, but obviously we could not find any deformation if we fed the network with only the tails. So this was really against our hypothesis. So then we deleted only the tails of the fish to see what happened. And when we fed the network only with fish heads, we could all of a sudden classify them as treated or untreated again. Not 100% sure, but fairly well. So this really confused us, that it worked with the heads, but not with the tails. And then I ended up on the train ride together with some zebrafish experts, and I brought out my computer and I asked them to see what they could see in the images. And then it turned out that apart from the bending of the tails, there's also change in sort of the back of the head. The drug also affects the brain or the backside of the brain of the fish. And if you think of a convolution, a convolutional filter can very often, very easily detect a change in volume. Of course, a convolutional filter can also detect the bending, but the change in volume would be a much easier thing to detect. While our visual system is really, really good at seeing if something is straight or bent, but we're not so good at seeing differences in volume because that's part of, or area, because that's part of our perspective. So the convolution picked up something that a convolution is better at detecting than what is visually more obvious. So to conclude, using this kind of simple ablation with photoshopping, the deep learning could really find features that we were previously not aware of. We had not noticed this change in the back of the head, but this is really what the network could point us to. And I think this is really one of the powers of neural networks, that they can detect something that is not necessarily visually obvious. We used manual sort of deleting parts of the images with Photoshop, but now there are much better approaches for really knowing which pixels are most significant for decision making. These are some examples of what's now called explainable AI. And here the color coding shows which pixels are most relevant for decision making. So for these cats and dogs, here it's obvious that it's the face and the nose that's relevant for classifying these animals as cats. But then in some cases, it's really a bit odd what parts of the image are most relevant for the decision making. Here, the face again is what decides these are the dogs. But then here in this image, it's something in the lawn in the background here that is most relevant for the decision. And here is a cat incorrectly classified as a dog. In this case, it's something in the fur that is probably then seeing some kind of dog pattern. And again, here, this dog is incorrectly classified as a cat, maybe because of this cat-like bending of the back. And so these are the kind of methods that are used today. And this is an example from a oral cancer screening project that some of my colleagues are working on, where we're training a network to decide based on these mouth scrubs if the cells are malignant or not. And here again, it's the color coding that tells what part of the image is most relevant. And here, of course, it's a good sign if it is the nuclei and the texture in the nuclei that are the most significant for the decision making, because that is what makes most sense to us. So, in our early experiments with these networks, we wanted to minimize size of networks, and we wanted to have simpler networks in some kind of approach to better understand them. And one thing that I wanted to mention that we realized when trying to simplify our networks is that the more a priori information that we can feed the networks with, the smaller they can be and still produce a good result. And what you see here is E. coli, little bacteria that we have in our guts. And the goal here is to segment out all the bacteria and then track them over time to follow their division patterns and their proliferation in response to different treatments. And we did approach this problem with classical methods first. And when we approached it with classical methods, we realized that what really makes the difference or what really separates the individual bacteria is this change in intensity. And then we realized that these handcrafted features, in this case wavelets and eigenvalues of Hessians, could really enhance these regions that were relevant for the segmentation. So we had realized that this pre-processing of the images made it easier for the forthcoming classical approaches for segmentation. So then when applying deep learning to this, in this case a UNet, we realized that we could really simplify the networks by including this pre-processed eigenvalue image together with the raw data to make the network learn faster. And with really trying to push for a more simple network, we see that if we did not include the pre-processed image, we got something, but if we included the a priori information in form of this eigenvalue image, we could get a much better result with a simplified UNet. So we could really improve the performance by doing this feature augmentation and seeing what is the information that we can provide ahead of time to really reduce the number of parameters and make the network smaller. But what I also wanted to mention with this particular project is that when we approached it with the classical approaches, we wanted to have a good way of describing the bacteria. So then in our segmentation approach, we included information about the length and the size of the bacteria that we expected to find. But with the deep learning approach, we only included then this eigen image and the raw image, but no information on the size and just shape of the bacteria. And with the classical approach, when we had the information of size, shape, and curvature, we realized that some bacteria that had odd shapes and sizes were missed. But really the power of the AI was that these bacteria could still be found because we had not added these restrictions. So compared to our previous approaches, when we built the whole segmentation on a priori information on cell size and shape, the AI approach could really find unexpected shapes and sizes, just because we had not put these restrictions in the analysis. And I think that's super powerful, because if we had done these large-scale experiments with our classical approaches, we would never have been able to find these super long bacteria. This is called snaking, which is one of the effects of or responses to antibiotics treatment, that the bacteria don't die, they just stop dividing, but they continue to grow as a way of sort of overcoming the presence of the antibiotics. And this forking, we don't really know what it is, but it's also quite interesting result of treatment. So those were some of our earliest projects that we did. Now I'm going to move over to digital pathology, which is a super fast growing field. And if you look at the bioimage informatics, sort of the community of people working with image analysis, it has been a very open community. People have been sharing algorithms, sharing data, and talked about all their different challenges and solutions. And there's a huge amount of free and open source software. I will provide you with some links towards the end of the talk. But with digital pathology, it's different, because this is a field where people believe that they can make money, and that makes people a lot more protective about what they create. We have approached the field, and we have seen the competition in it. But I'm going to tell you our story, and I think it is quite similar to a lot of other approaches. So then, what is digital pathology? This is kind of to make you wake up a little bit. This is the kids' game, Where is Wally? And if you look in this image, I'm hoping that you can all find Wally somewhere. If anyone wants to be brave and say where he is, I'll be happy to take your suggestion. Maybe you're all still a bit sleepy. Anyway, Wally is located right here. But he has also some friends spread out in the image that have that look similar, but are not quite looking the same as Wally. And why am I showing you this image? Well, I'm showing this to kind of illustrate the problem. Because now if you imagine that you were trying to find Wally looking under a microscope, and you were under time pressure because you had so many of these samples to look at, and someone's life would be dependent on whether you find Wally or not. And this is what life is very often like for a pathologist. They have to look at these samples to see if there's malignant changes, for example, if there's cancer. And you can very often end up in situations where you make mistakes and miss things. And if you think of it, this is a biopsy of a prostate. So it's a thin needle that is stuck into the prostate, and then the cells are drawn out, and this is like a really super thin sausage that is then sliced a long way and then put on a piece of glass. And to find the malignant changes, you really need to zoom in, and you need a large high definition screen simply to view the full sample at high enough resolution to find these malignant changes. So this takes time, and this is where decision support or guidance by AI has become very, very popular. And I'm going to tell you more of the details about this story, but I'm thinking that you probably want to have a bit of a break. So I think this could be a good time if you have any questions, and then we will take a 15-minute break until 9 o'clock. And then I will continue with the story about the AI for digital pathology after the break. So do we have any questions at this time? So we were at this point of looking into decision support in digital pathology and how to automate the detection of malignancies. And as I said before, this is a field where a lot of research is going on. And the way this actually started for me was that I had not planned to work with digital pathology, but I ended up talking to my next door neighbor, you know, chatting about life in general. And then all of a sudden my neighbor said that, hey, you know, I have 7,000 H&E stained prostate biopsies where malignancies have been manually marked and graded. Do you think it would be possible to do anything with image analysis? And maybe not the usual thing that neighbors talk about, but we had, you know, a common interest in research. And when I heard about this, I got quite excited, because this meant that there was a lot of high resolution image data and really with labels. And this is the key thing for creating good deep learning. And so what it was that there was physical pen marks on these slides next to the regions where the tumors were. So this pathologist had looked really carefully in the microscope and then taken a pen and made a pen mark saying, next to this pen mark, that's where we have the cancer changes. And I was lucky to also have good students around that could engage in this. So, and also one thing that was really nice was that on each of these glass slides, there were 2 slices of the same biopsy. So one slice here and one slice here. So showing parallel sections of the same biopsy. But the pen marks were only on one, meaning that we had, you know, a clean piece of tissue here that was not annotated in any way. And then what we had to do was then to train a network. And the first thing was to do was then to use more classical approaches to figure out what parts of these biopsies to cut out and call malignant and benign. And what we could then sort of digitally transfer the signal or the pen mark to the tissue and then cut out lots of these little patches. We actually cut out 5.1 million patches that we could use for training. Each of them corresponded to 540 by 540 micrometers, a quite small region in the tissue. And we actually ended up reducing the resolution of the images. It turned out that we tried lots of different patch sizes, but in cancer grading, you need to see a little bit of the context. You can't have 2 small patches because then you're too close to see the surrounding pattern. So we were playing a bit back and forth with this and then came to the conclusion that lowering the resolution and having fairly large patches was what worked best. And we ended up building a deep neural network and ensemble, so a collection of many networks. And we used the Inception V3 model that was pre-trained on ImageNet. You might want to question why you would want to pre-train it when you have 5.1 million patches to train, but that's where we started. I don't know, I think it would have given equal results if we had started just with not a pre-trained network. And then of course we got then the 30 answers out for each patch. And then we had many patches per biopsy. So then we combined the results from these patches using boosted trees to get a final result. So the network actually consisted of 2 parts. One that decided if a patch was benign or malignant, and then a second network that decided what grade or how serious this cancer was. And then we could use it on unseen or unlabeled biopsies and apply it. And then we got like a heat map as output that we could then overlay on the original image and use it as a guide for the pathologist. But as always, you want to know how well does this really work, and can we trust this? Is it any useful? So we did evaluate it on an independent data set. And if you look at this ROC curve, we have an area under the curve of 99.7. So it's really, really good at distinguishing between benign and malignant biopsies. And it worked equally well on a completely new data set, or almost as well on a completely new data set. And what we can see here is that if we have, this whole box of many, many slides that have to be graded, if we would apply this decision support, we could dismiss as many as 80% of the negative cases without missing a single cancer case. And for all the cases that we would actually look at, we would then have this guide to see where to focus our attention. So we could really speed up the whole process and really not miss many, or hardly any cases at all. But then again, does it really work better than the visual grading? And in this case, we were lucky to have access to actually a whole consortium of prostate experts or uropathologists. And they had a collection of 87 difficult cases. And these 87 difficult cases had been graded by 23 persons. And not everybody agrees. And one of these 23 persons was also the person that trained or had made all the pen marks. So it was our AI trainer. So then to really see if our AI system was as useful as a pathologist, what we did was to compare all of them against one another. And then we, this visual grading and deciding where it's cancer or not, it's difficult to say what is right and what is wrong. But if we have the hypothesis that the more people that agree that there's cancer, then we can be more sure that there's cancer. So when in comparing these, we would say that whoever agrees with other experts more often is better at doing the grading, but those that deviate a lot are maybe not as good as the grading, which can of course be discussed, and I will kind of come back to that. So we did this two different experiments, one where we looked at grading or putting the samples correctly into each of these 5 levels of grading, and then also one where we graded, grouping it into the lowest grade or the grade 2 and 3 or 4 and 5 together. And when doing so, we realized that their AI system was, in the second case, it ended up on place 17 out of these 23. So doing okay in comparison to the other uropathologists. But it was really important to remember here is that these uropathologists are really the best ones in the world. And then if you compare it maybe to the rest of the pathologists that would be available in other places in the world, maybe one can conclude that our AI system is good at grading, is as good as grading as these really internationally acknowledged uropathologists, and that it does have a value in this decision support, especially in places where there is a lack of experts. We could really apply it to guide, and maybe also as a system to help those that are less experienced to see where to look more carefully to make a final decision. And this, as everything else that I've presented, is available in publications also. But what I want you to see here is that when we did this comparison, it turned out that our AI trainer was actually top ranked. So a person that really had a huge amount of experience in doing this kind of grading and agreeing in many, many cases with other international experts. And this sort of illustrates the drawback of this whole story. Because our training of this AI system that worked so well depended on these 7,000 H&E stained prostate biopsies that were manually marked by this really professional uropathologist. And we could see that not all uropathologists agree. So having access to this really high quality data is very rare. And if you think about it, the only thing that we showed with this network is that we could repeat what a human could do. And I mean, that's of course nice, but it's not super exciting. I think the goal should not only be to replace humans, but really to do something more than a human could do. So we really want to go beyond the human ability to grade these cancers. And that's what I'm going to tell you about next. So if you think about analysis of tissue samples, what you would typically do if you have a suspected tumor, it would be to cut it out. And you would do this preparation and put it on a tissue slides and then either just look in a microscope or digitize it and use an automated approach like I just presented. But this is a bit limited. We only look at the size and shape, but we don't really know exactly what kind of molecular changes happened inside this tumor. And it's really the molecular changes that we would need to know if we were to design a good treatment of potential new tumors or malignancies in this patient. So what people have been doing for many, many years is to do a genetic analysis to see which genes are active in these tumors. But what we would usually do would be to take sort of a piece of the tissue and put it in a mixture and get an idea of the combination of genes that are expressed in the tumor. But there is a lot of heterogeneity. Different things are happening in different parts of the tumor. And by taking the bulk like this, you don't really know what is where. And you don't know which types of cells have been changed. But we now know that there are many different cell types involved in cancer. And if we can then sequence and look at the gene expression of individual cells, we can know what are the different cell types. Here, each cell type is represented by different fruit or berry. So we can then understand what combination of cells are present in the tissue. Is there, for example, a strong immune response against the tumor? Can we boost this to kill the tumor? But with this approach, we still don't know where the cells are in the tumor. And here we have a new method that's called in situ sequencing or spatial transcriptomics, where we can do the genetic analysis, but see it in the spatially resolved tissue. I'm not going to go into details of this, but it is a really cool method and was selected Method of the Year in Nature Methods for 2020. Our version, there are many different versions of it. Our version is called in situ sequencing. So you can know where in the tissue different things are being expressed. And what you get out is that for each position in the tissue, you can see what genes are expressed. So here you can see different patterns. This is a slice from a mouse brain, and you can clearly see different patterns for the different genes. And then this can tell us what is actually happening in the tissue. So if we look now again, this is not prostate, this is breast cancer. And you can, in this case, I think, see visually that up here and also down here, you have like fluffy cell nuclei, while the nuclei that are here and here are more compact. So if you would manually annotate this, you would probably say, okay, here is the cancer, and here's the cancer, and here we have normal tissue. But then we might have problems deciding where the borders go, and if all of us would do it, we would have slightly different results. But then if we do this in situ sequencing, we can overlay this and then have the sort of the molecular information tell us where there's tumor and where there is not tumor. And then we can use this as input. So instead of having the pen marks by the pathologist to train our AI system, we would sort of take this fruit pie representation where we have the molecular information and use that to train our AI system. And this is something that we are working on now. The tricky thing is that this approach is quite expensive. So we need to find a smart way to creating these large amounts of training data. And one thing that we have been trying just to see the potential of this is to go back to the network that I just presented for the prostate cancer grading and sort of reuse it, not use it for classification, but use it as a feature extractor. Because you know that the last layer before you make the decision is essentially a lot of sort of condensed information describing the morphology in the tissue. So if we take the features from the last layer of the network, and instead of making a classification, we take this feature space and reduce it. In this case, we used a UMAP for model aggregation, and we can present this output as a in 3 dimensions as a color map. But we can also cluster this feature space. So we decided to cluster it in a 10-dimensional space, and then just reduce it to a small number of clusters. In this case, we reduced it to these clusters and then projected them back to the tissue. So these are clusters that were created really without any a priori information on what's going on in this piece of tissue. We only clustered based on the features that were designed to detect malignancies, and we get this pattern. And then we compare that to a manual annotation by a pathologist. And now we color-coded these to sort of enhance the similarities and differences. And we see that the pathologist has slightly more detail, but the general patterns are repeating. And what we also noticed here is that we decided to do a larger number of clusters than the pathologist. And then you see that this pink part here is the same at the lower and the upper part. The pathologist has given the same class to these 2 parts of the tissue, while the unsupervised clustering by these features extracted from the network that were trained to identify different grades of cancer gave these 2 clusters 2 different colors. It said there's a difference in what's going on in the upper and the lower part of the pink cluster. And then we could go back and look at the gene expression information and looking at many different genes and combinations of genes. And it might be a bit tricky to see here, but this piece is the same as this and this piece. And as you see, there is one gene that is more strongly expressed in this lower part, and another gene that is more strongly expressed in the upper part, showing that there is a strong correlation between differences in morphology and differences in gene expression that sort of go beyond what could be visually seen in the tissue. And this is an ongoing project. We haven't published it yet, but we're in the process of doing so. So it's really the fruit pie kind of representation that can tell us more about how correct or how wrong this manual annotation would be. So now for the next part of my presentation, I wanted to comment a bit on this sort of drawback with deep learning, in that people say that it requires enormous amounts of manual annotated training examples. And it doesn't have to be manual annotated. It could be annotated by different approaches, especially in microscopy, where you can have different tricks of overcoming the need for manual annotations. So one thing that we often want to do is to follow living cells to see what happens over time. The tricky thing with looking at living things is that we don't have so many stains that we can apply, because stains almost always influence the cells, and they get sick from the presence. And also, if you use fluorescent stains, just the fluorescent light in itself is toxic. So it makes it difficult to follow living things. But at the same time, it's difficult to automatically detect individual cells if you don't have a stain. So we wanted to have an AI system here, but we didn't want to sit and manually annotate lots and lots of cells. So what we did was to follow the cells over time, and also collecting these stacks of images so that we would sort of change the focus level and having more information per cell. After doing the time-lapse experiment, we fixated or sort of killed the cells and added 2 fluorescent stains, one staining the nuclei and one staining the cytoplasms. And here you can imagine that it would be super easy to use a classical approach to find individual cells, because they're super bright against a dark background. And that's exactly what we did, using Cell Profiler, which is this open source image analysis software that is quite widely used. So then you get the labels of all the cells. But these labels then match the last time frame in our cycle of unstained cells, meaning that now we have the ground truth for the last cycle in our time lapse. This means that we can combine these two to train a network to detect cells also that are unstained. And once we have trained that network, we can apply it to all the previous frames in our time-lapse experiment. And rather than getting, a clean cell segmentation, what we get as an output is a probability map showing where it's most likely that the cells are located. So this almost looks like a simulated image or simulated fluorescence image. And again, this image is much easier to segment than the one that we started out with. And we can now identify our individual cells and then track them over time. So we learn the cell outline from our fluorescence data. In a similar way, when you work with fluorescence, you are limited by the spectra of both the excitation and the emission of the fluorescence. So you cannot not have an unlimited number of stains at the same time. But if you imagine that you wanted then to expand the number of stains, but still extracting as much information, you could see if there's any of the stains that you could sort of learn from the other stains. And what you see here is a nuclear stain. And on this side, you have a lot of other stains, but no nuclear stain. But as you can see, it's kind of easy to still imagine where the nuclei are located. So again, we can train with this as a segmentation mask, and this is the input. and then learn where the nuclei would be located. So we can sort of learn things from one another, and in this way free channels so that we could use the blue color that is usually used for the nuclei and stain something new and then extract more information from the images. And this is something that we used more recently also. So it should say adipocytes here, it should be a P there, sorry about that. So this is also a challenge that AI Sweden organized together with AstraZeneca. And what AstraZeneca wants to do is to study fat cells. So what you see here are two images of fat cells. And to the left, we have a fluorescence microscopy image. The green are the fat droplets, the blue are the cell nuclei, and then we have a red stain to show the cytoplasm. And with these stains, it's fairly easy to automatically count fat droplets, count nuclei, associate fat droplets with nuclei, and then count fat per nucleus. But the problem is that the stains are toxic and expensive, and we cannot follow the cells over time. If we instead image the cells without any stains, with just regular light, we have an imaging system that is both cheap and non-toxic to the cells. We can take cells then directly from a patient, and we can follow them over time. And we can, for example, see how the cells respond to different treatments. So we would really like to do our experiments here, but we know that it's difficult to extract features from these images as compared to these images. So we kind of want to do this imaging, but we want to use this for measuring things. And that was the challenge in itself. So then can we create an image like this if we have a image set like this with a cheap non-toxic staining? And we approached this with actually 3 different networks, one for each of the fluorescent channels, trying to really maximize the use of the information. And one of the things that we knew would be used for evaluating the performance of the network was this type of traditional cell profiler-based approach, where you would start by finding the nuclei as a seed for cell segmentation. So we knew that the segmentation of the nuclei would be really crucial for the output. But as you can see in these images, it's really difficult to see where the nuclei are located. So when training this, we did not only use the fluorescent image as such as input, but also a segmentation result of the image, so that we had some like privileged information, and also used both the image losses and the segmentation losses in our training. And also some specific tweaks to how to make the most of the information for the fat droplets and the cytoplasms. And in the end, this is the same image as I showed in the introduction. So this is a real image, and this is then the artificial image produced. This real image was not used for the training, so this is the result on an independent training set. And my students that were doing this, they were working super hard, and they were super happy to also win the first prize in this challenge. And the description of the approach is now under publication. And hopefully it will also come to use for drug discovery. One thing that we have also been combining with deep learning is confidence metrics. Very often in microscopy, you can have really big images, but the information that you care about is really sparsely distributed. So in this particular problem, the question that we want to ask is where in the lung a drug goes. So this is an inhaled drug, and the purpose of the experiment is to see that the drug ends up in the correct part of the tissue. And the parts of the tissue that we care about is actually two of the very, very thin cell layers in the epithelium of the airways. So there are huge parts of the image that we do not at all care about. What we really want to do is to look at highest possible resolution in a very small part of the image. But then in order to figure out where this small part of the image is, we then work with multiple resolutions. So first we really just subsample the data a lot to see where the airways are located. And then we go in stepwise to pick out these regions at then high resolution, and then continue our search at this high resolution. So this is the, if we then focus on what we get out at the highest resolution, this is where we get. So our output then for detecting these different parts of the tissue is a map like this, saying that we have found an airway, we have found the subepithelium, and the also some blood vessels and some alveoli. And the thing that we really want to study is how much of the drug is located in each of these different regions. But in part of the data, we might have made mistakes. You see here we have a bit of the yellow and the red class. And here we have a bit of the orange class really close to the airway, so that might be an error as well. But then if we can, in our network, also include a confidence metric, we can select to do the analysis only in the parts where we're most confident about our classification result. So by having this confidence included in our classification, we can reduce our regions to only the most confident ones, and then get a much higher detail in our description of our drug response. So if we include all our regions and then compare drug response, we don't see so much of a difference between these different parts of the tissue. But if we reduce our regions to only the parts of the tissue where the network is most confident, we can get a much better discrimination and much better see exactly where the drug is located. And also by using the sort of multi-resolution approach, we can, rather than analyzing the full image in the highest resolution, we can reduce as little as 5% of the total image data and sort of throw away things that are less interesting as we go along. I just wanted to show you this also. I hope the video comes across also on Zoom. What you see here is in each of these boxes is a different set of cells. And you see some orange stuff flying in. And that is lipid nanoparticles. That's the exact same thing that is used in many of the corona vaccines that we use today. So it's inside each of these little particles is a RNA molecule. And whenever the RNA is taken up by the cells start to produce, in this case, a green stain. In the case of the corona vaccine, it would be the spike proteins. And in order to understand this process, we are working with this type of time-lapse images and then a deep learning approach to see if we can predict what morphologies of cells that lead to this green fluorescence. And the goal of this whole project is to better understand how to best design these lipid nanoparticles to get the best possible uptake of the drug. And if you want to read more about it, there's a link here. So it's a time-lapse information that we incorporate in the networks. And in general, when you work in microscopy, one of the powers of deep learning is that you can sort of play tricks with the data, and you're always kind of fighting against trying to overcome complicated imaging methods and toxic imaging approaches to enable a more natural environment for the cells and enable cheaper imaging and the possibility of scaling up imaging. And one area of cell biology that a lot of people are studying is traction forces and how cells sort of move across the surface. And the reason why you want to understand cell motion is, for example, in cancer, to understand how cells spread in the tissue, but also in wound healing and other developmental processes. And the classical approach to study these traction forces is to grow the cells on a surface where you have lots of little flourishing beads. And then as the cell is pushing and pulling, you can see how the beads move. And then from the bead motion, you can analyze the traction in the cell. But we had then access to this force data, and then also an image of only the cell by itself without the force data. So the force data was calculated from these beads. And then with this type of pairs of images, again, you have the possibility then to learn one thing from the other. So using these pairs of force data and raw data, we could predict the traction forces so that we can scale up our experiments and study traction forces without the need of the force data or these fluorescent beads. So this also kind of shows the power of deep learning to overcome complicated imaging systems by sort of simulating their output. I think one of the final things that I wanted to bring up is some of the drawbacks with deep learning. I showed you before that with the zebrafishes, we thought that we were building a system that could detect the bending of the zebrafish tail, but instead the network actually found the change in the head of the fish. And in that case, it made us discover something new. But this is not always the case. And sometimes a network learns something different than we intend for it to do. And this is an experiment that made us realize just that and showed us how careful you need to be. And in this case, we were working with electron microscopy. So electron microscopy has very high resolution, meaning that it's also very sensitive to shaking. If something is moving slightly, every little motion will be picked up when you try to do the imaging. So if you want to image a larger area, you need to position the right area that you want to image under the microscope. And then you have to wait a little bit for every motion induced by moving the sample to sort of reduce to get a nice and crisp image, which then means that it takes time. So in order to scan a larger sample and sort of figure out where you might have regions of interest, it takes an enormous time if you want to stop at every place. So then our hypothesis was that we might be able to sort of scan quickly and collect series of images with motion blur, And then if we could if we could model the motion blur, we should be able to go back and recreate the still image, and in that way gain time. So we had two different data sets. One was this data set of what's called a calibration grid. So it's a pattern that is human-made and well-defined, and that would be easy then to quantify how well we can correct for the motion blur. And here you see the motion is in this direction, meaning that the lines that are perpendicular to the direction are really blurred. We also had images of a kidney sample, and here we have a completely different resolution. The shape that you see here, that would be a single cell nucleus. So these are really high resolution images. And what we care about here is different, really tiny structures in the inside the cells. And here you see then the blurred version of this kidney. So the goal was to see, can we learn this from this if we have them the stack? And we tried many different things, but what we realized was that there were artifacts. So then we decided to really evaluate what these artifacts were using a set of synthetic images. So we only trained, this is a bit strange sounding maybe, but we only trained on real data. And then we evaluated on synthetic data. Usually people do the opposite because the synthetic data is where you have the control. But in this case, we really wanted to have control of the evaluation. So we created these 3 synthetic images, and then we added synthetic noise and motion blur to sort of make them look like what happened when we had the motion blur in the microscope. So you see these parallel lines are spread out and the round dots are a bit elongated. And then we tested several networks, but here we focus on the one that performed the best. And then if we only trained on the kidney data, we got this output, which is still a bit blurred, but it looks okay. But what was really the most interesting part here is that when we trained on the calibration grid data, on this data pair, what we saw was that the network introduced extra lines in our images. You see there are stripes here that are not at all present in the synthetic input data. When we took a network that was trained on both kidney data and calibration grid data, we could get rid of those lines. But what's the sort of take-home message here is that the network did not only learn the noise and motion blur that we intended it for it to learn, but it also learned the texture and the structure in the image sample. And that's why the network that was only trained on the calibration grid sort of tried to introduce a calibration grid type of pattern in the output image. So it learned more than we wanted it to learn. And that is, I think, super important to understand when you work in biology, so that you don't somehow think that there is a structure there that is actually not at all present. So if we go back to where we started, sort of with all these downsides of deep learning, what I want you to take home from today's lecture is that training a network really relies on high quality training data. And it relies on reliable manual annotations that often requires rare expertise. And this is also not always 100% reliable. But what is really important is that the training does not always require manual annotations. There are many ways to come around it. There are in many cases where you can have alternative staining approaches or alternative imaging approaches that can provide the guidance for training instead of manual annotations. What I also want you to remember is that a network may not always learn the same features as you regard as relevant for classification, and that a network may discover something that goes beyond the a priori human knowledge, or it may introduce something that is not really there. And this is where you need to be super careful with your evaluation and really question what you see and try to understand what you get out of it. And also, finally, there's a lot still to be tested and learned and discovered in this field. So you have plenty of things ahead of you that you could potentially dig into. And really, an AI is probably much more of a gift box than a black box. And I believe that AI can really help us find new biomarkers and new diagnostic cues, and also maybe help us find new treatments. And with that, I would like to say thank you. And again, also a thank you to my research group who has been involved in all these different projects. So with that, if you have any questions, I will be happy to take them now.